Hello, welcome to DevoWorm. Today we are not going to have an in-person meeting. I'm giving an update on some things that we have in the group. We have new data sets that I'd like to share. These are secondary data sets from different data sources, so I'm going to go over those. And then I'm going to go over some papers. So the first thing we have are these larval data sets. So in last week's meeting we talked about larval stages for different organisms. Of course, we have larval stages in C. elegans. We have larval stages in other organisms. And we have some zebrafish larval data that we've been working with for a couple of years in the group. And we have some of those analyses published, at least in one paper. And then we also have some new data from C. elegans L1 stage. So L1 is the first larval stage right after hatch of the egg. This is actually well mapped to both the adult and the embryo, so let's take a look at some of these data sets. So this is the C. elegans L1 data set. This is the first larval stage, so we have a list of cell names, and those cells are adult cells. So these are the cells that result from development. They've differentiated at the tips of the differentiation tree, and now every cell in this are considered adult cells. So what we have here are adult cell names. We have their XYZ position. We have a normalization of that. And then we have some intensity data which have to do with gene expression and some other things here, color information for those measurements, and then a mass, which we typically don't use. But this is basically the data you want. So you have the names of the adult cells. And of course we have an adult C. elegans model, which we can map these cells to. So you can look, for example, at these cells in larval development versus the adult. And then we also have data on cell position for developmental cells in the embryo. And because there's a deterministic mapping between the lineage tree and the adult cells, we can actually overlay an embryo with an L1 point cloud. So all of these positions, if you plot them out, they yield a point cloud, and that can be basically it's the shape of an L1 worm, and that can be mapped to an embryo as well. So you can get information about what's going on with that corresponding developmental cell in the embryo and its position, and information about migration and other things. So this is also good for training machine learning models. So we have, a good data set here, several thousand cases. So that's going to be of interest, I think, to people working on different problems in C. elegans. Then we have two zebrafish data sets. So we have cell larval data sets and this scRNAseq data. And so the cell larval data set is similar to the data set we have for C. elegans L1. We have actually two stages in zebrafish. We have the zygote stage and the cleavage stage. So these are just different stages of development of the larva in zebrafish. And so we have here a cell ID number, which is kind of, if you go back to the original data set, and I'm not going to show that here, but there's an ID for the cell. At least if it's a unique cell or not. You have this position data, and then you have a timestamp, which is this minute data. So basically we can go through and we can plot this out in time, we can plot this out in space, and we can get an account of what the zygote looks like, the shape of that point cloud, what that should look like. We also have the cleavage stage, which is a little bit later in development. So the zygote is early, the cleavage is a little bit later. Again, you can see that it starts at 46 minutes and it goes down, I don't know how many cases we have here, but quite a few. And we, of course, have the unique cell ID and then XYZ coordinates. And, of course, we can do things like look at the zygote versus the cleavage, look at the cleavage stage or the zygote stage on its own, or even compare these to C. elegans in some way. So it's very nice data. It's all these point cloud data these positions describe a centroid of a cell, and that gives us a point cloud, which we can then do things with. We can also use these data to train machine learning models and this sort of three-dimensional shape discovery. So we also have the scRNAseq data, and this is also for Zebrafish. So scRNAseq, it's this next-gen sequencing technique, and in this data set, they've used this to, kind of build these maps, these visualizations. And so this is where we have, these are numeric data that, you have to go back and look at the original paper to know kind of what's going on. But basically, we have data for the wild type cells, and we have two variables here. This is going to yield a t-SNE map. So these are the t-SNE components that if you plot them out, you'll get this standard t-SNE map. Then we have this developmental landscape from DropSeq. So this is actually, these are XYZ coordinates, and these are the same. I think these are oligo sequences that they have, and these are from the wild type. So you can go through and look at those. So they're probe IDs with the cell ID, and then you have 3 dimensions. So you plot this on a 3-dimensional map, and you get this landscape of these cells and the sequences that are being expressed in those cells. Okay, that was a little bit about the new data sets. To find out more, we'll have to go into the GitHub repositories and look at these data sets where it has the reference for the original paper and the CSV files. We always work from CSV files. Those are the simplest way to do a lot of these data. So let's move on to the papers now. I think the papers are interesting this week. We have a series of papers on generativity and development. That's going to be very interesting. We also have this paper on transcriptional accounting, and I'll show you what that is in a minute. So this paper is called Transcript Accumulation Rates in the Early C. elegans Embryo. So this is from the group of John Murray, who of course has generated a lot of the cell tracking data for C. elegans embryos. And so he's doing this kind of work now with these two collaborators. And so what is transcriptional accumulation rate? Well, that's what we're going to talk about. So dynamic transcriptional changes are widespread and rapidly dividing developing embryos where cell fate decisions are made quickly. So what they're saying here is that you have an embryo, you start with a single cell, the cell divides, but of course at the same time genes are being expressed in that cell, and there's a transcriptional complement of our mRNA, and there's an mRNA content in that cell. And as the cell divides, that mRNA segregates into one of two cells. It could segregate into the daughter cells evenly or unevenly. And so this is what he's talking about with respect to, these transcriptional changes, and you also have dividing cells, rapidly dividing cells in an embryo. The C. elegans embryo overcomes these constraints partly through the rapid production of high levels of transcription factor mRNAs. So in the embryo, because the cells are dividing so quickly, there's a lot of mRNA being made. So at a very high level. And so, we think about gene expression in adult human cells or adult mouse cells or whatever. And there are these experiments you can do where you can actually turn off the transcriptional machinery with a drug treatment. And you can watch the decay of mRNA over time. And it takes maybe 20 or 30 hours to completely deplete. And of course, the cell dies. So, there's a transcriptional rate you can establish. How long does it take to, kind of make mRNA, and how big is that complement within the cell of a certain mRNA? So, this is something you can approach quantitatively. And so in C. elegans embryos, because you keep dividing and you need to have enough mRNAs to do what you need to do, but you also have to keep fractioning it away to put it into your daughter cells. You need to have this high level of synthesis. So this is also important for cell fate decisions because you need to have things upregulated at the right time. They need to be available for you to use as a cell. So these are all, things that involve mRNA pools within the cytoplasm, and the ability to utilize that pool when needed. Transcript accumulation rates for some developmental genes are known at single cell resolution. So we know kind of what these accumulation rates are. In other words, transcription proceeds at a certain pace, and there's an accumulation of transcript, and there's a certain amount of time that the cell has to make it before it divides into daughter cells. And so if the daughter cell is going to differentiate one or both of the daughter cells, you need to be able to produce enough transcript and accumulate it so that that daughter cell will have the transcript available for it to use. So in the past several weeks, we've talked about cell division, we've talked about cell cycle, we've talked about how you need certain amounts of mRNA or certain amounts of gene expression to occur before you can move to the next stage of cell division, or sometimes you need to shut off mRNA production to proceed to the next level of cell division. And so because of those, transcription accumulation rates are very important, whether how long transcription goes on for, what the rate is, and what that accumulation is in the cytoplasm. We also talked about, I think it was last week or the week before, about the speed of transcription or the speed of cell division. And so the speed of cell division really is about the speed of transcription and the accumulation of transcription. So that's something to keep in mind, that this is closely linked to cell division. But this is also closely linked to cell differentiation. So we know some of these transcript accumulation rates. We can do experiments to quantify these. But genome scale measurements are lacking. So we know kind of what some of these are for certain developmental genes, key developmental genes, and maybe that's all we need, but we don't have these genome scale measurements. We don't know across the genome what's going to happen. And so that may be important because, sometimes you have, all sorts of transcripts that are needed for sort of to keep a cell viable or to allow it to differentiate to a new state. We've talked about genes needed for differentiation, but we're only really kind of interested in the genes of largest effect. Sometimes you're going to need other genes of lesser effect to, the transcripts of those genes to be there in order to differentiate properly. So it would be nice to know all the details of that. We estimate zygotic mRNA accumulation rates from a single-cell RNA sequencing data, calibrated with single-molecule transcript imaging. So they're able to do this kind of imaging where they kind of get these, they establish these accumulation rates from the single-cell data. So we just saw some single-cell data, but we're going to do, I think, a different method in this paper. Rapid transcription is common in the early C. elegans embryo, with rates highest soon after zygotic transcription begins. It's interesting that, we have this very early establishment of very rapid transcription. And of course, it seems like it's right at the sort of the root of the lineage tree. So as soon as you sort of start to divide into two cells, that's when you have the fastest transcription. And then it kind of tails off after that. But, in this early C. elegans embryo, you get this rapid transcription. It's very common. And so that makes sense. If you think about what's going on in the early developmental lineage tree, you start off at the one cell, you divide into two cells, you establish your basic polarity, so you have your AB lineage and your P1 lineage. And then in a couple division events later, you end up with your 8 founder cells. Those 8 founder cells are sort of key in the establishment of sublineages that will turn into different tissues or, be different regions of the embryo. And so it's important to sort of get all of the transcripts in place to get a large pool of transcripts. And as we said before, transcripts decay at a certain rate, but they don't decay that quickly. mRNA decays around, maybe the 12 to 24 hour period. So, the half-life is what I'm talking about. So if you start in early development, and in the C. elegans embryo, our development is, not that long, you end up with, being able to preserve a lot of that early transcription throughout the life of the embryo. So by the time that we get to, a lot of these cells being differentiated, maybe some of that mRNA sticks around. But we definitely have enough where, the rate of division is greatest. So we can get to those differentiation events and have the genes, or at least have the mRNA complement at the right amounts to make a difference, to support that differentiation event. Okay, so high-rate genes are enriched for recently duplicated sulfate regulators and share common genomics features. So, but he says high-rate genes, that is where you have this high rate of transcription. This is enriched for recently duplicated sulfate regulators. This is very tightly linked to sulfate and for, duplicated regulators. So this is, maybe important for cell duplication. In our new paper, we speculate about how, cell duplication might be important for differentiation, how, we have these specific genes we're looking at for sulfate specific things. So this is maybe an important thing to look into with respect to what we were talking about with differentiation waves. We identify core promoter elements associated with high rate and measure their contributions for two early endomesodermal genes, CEH51 and SDZ31. So these two genes are involved in endomesodermal sorts of things, so early tissue specification. Individual motifs modestly affect accumulation rates, suggesting multifactorial control. So this is where we have motifs of genes and how they affect accumulation rates. So that means, I guess, that genes have to be regulated together, and this is something that we, maybe there's some sort of multifactorial control. These results are a step towards estimating absolute transcription kinetics and understanding how transcript dosage drives developmental decisions. So this idea of dosage is this same thing as half-life. The dosage is the amount that's there. The half-life is the decay over time. So over a certain amount of time, you'll get half of that dosage, and then a certain amount of time later, you'll get half of that half of dosage. So it decays sort of linearly over time, and not always. Sometimes, especially when you don't shut off the transcriptional machinery, you get nonlinearities where you get these mass accumulations and then decay, and then maybe some more production, and so forth. I can also say that in mRNA decay experiments, we can actually force the cell to sort of adapt to its environment, so we can do certain things to the cell before we shut off the transcriptional machinery, and it shows sort of an accumulation of mRNA way over baseline, and then you get this decay. So there are a lot of things going on in the transcriptome and in this cytoplasmic complement. They kind of make the point here that absolute transcript accumulation rates, or the rate of change of RNA copy number that has led to a timely production of transcripts at the appropriate level, are rarely explicitly measured at a global scale during development. So there are some experiments that have been done on Drosophila where they've done quantitative imaging. They provided detailed measurements of the transcript production rates for some early patterning genes. So these are genes that are involved in morphogenesis, in striping, and so forth. One such study found the accumulation rates of the gap genes hunchback, cripple, knurps, and giant to be fairly similar, 33 transcripts per minute per nucleus. So this is where we have these genes that are involved in this, gap genes are involved in different pattern formation activities. that have very similar transcription rates. So this is 33 transcripts per minute per nucleus. So this is actually pretty useful, and this is, of course, over a 15-minute nuclear cycle. These rates are high, with levels approaching the theoretical steric limit based on RNA pull-to footprint. So this is where, the polymerase can only move so fast, and it can only produce so many transcripts. And so this is basically, there's a sort of an upper bound, and we talked about this in this article on increasing the speed of cell division for bioengineering. And so, we were looking there at bacterial cells. In this case, we're talking about Drosophila cells. But in both cases, you have this rate limiting factor of the polymerase. And so that's interesting that, you're able to get up to that theoretical limit. So this would allow for 1 pull-2 every 70 to 80 base pairs. And so this is something that we've gotten from live imaging of mRNAs, but there's some limitations to that approach, and so that's going to set up their approach using next-gen sequencing. And so they picked the C. elegans embryo because it provides an ideal developmental system to answer these questions. It develops with an invariant lineage where division patterns are highly reproducible across embryos, allowing equivalent cells to be compared between individuals. This robustness results in part from a high level of precision and expression levels, and this has been observed in intestinal transcription factors, these citations 11 and 12. The intestinal specification GATA factors, N3 and N1, accumulate to very high maximum transcript levels. And in this case, this is over 300 mRNA molecules per cell in short cell cycles. So this would be a 15 to 20 minute cycle. So this is where you have these two genes that are GATA factors. They're in these intestinal, what are going to be, intestinal cells. They're specifically intestinal transcription factors. So you go in the adult and check to see that they have this function, and they actually accumulate reproducibly, meaning over many animals, to very high maximum transcription levels. So I guess this is an upper bound for the number of molecules per cell that can be produced in about 15 to 20 minutes. Proper gut formation requires these transcripts to surpass a threshold level, emphasizing the importance of transcription rate in developmental progression. They want to measure high transcript accumulation rates and characterize these rates genome-wide. So they actually look at the read counts in scRNAseq data. So actually they are looking at the same technique, scRNAseq. So what you have in scRNAseq is you have these short sequences that you actually will try to sequence and then count the number of sequences of the same type, align them with the genome, identify what those sequences are, and then get a count of those common sequences. And when you have a lot of them, of course, that means that you have high expression. When you have very few to none, that means you have low expression. So this is what they're doing here. And so they're able to gain insight into absolute transcript levels in C. elegans embryonic cells. Genome-wide distributions of transcript accumulation rates vary substantially across cell types and lineages, but are highest at the 8-cell stage of development. So this is the 8-cell stage where you have your founder cells, and this is exactly what we might expect given what we know about the lineage tree. That this is when the sublineages are being founded, and so we expect this sort of transcript accumulation to be highest when we want to start our sublineages. High-rate genes tend to have more recent duplicates and are enriched in chromosomal clusters, indicative of a rapidly evolving class of genes. We identify gene features associated with rapid accumulation, including short primary transcript length and lower intron size and count. So this is where we get into this paper a little bit, and they're using Fish to normalize or to compare with the scRNAseq data. So Fish is a sort of an imaging technology where they can use fluorescent probes to validate a lot of what they're getting with the scRNAseq. So this is where they talk about how scRNAseq measurements can be noisy, and they validated everything on Fish. So this is where they get these oligo probes to target the mRNAs, and so they can actually visualize the mRNAs in the cells. So the idea is that if the fluorescent signal is brighter or greater in the SM fish, and the number of transcripts is high in the scRNAseq, then that's validating the technique, and it's sort of giving support, lending support to what they're finding with scRNAseq. So it's a nice design where they can actually kind of validate what they're getting. They're not getting, because what you get with scRNAseq is you just get a set of sequences, short sequences that are sequenced from bulk sampling, and then you have to map that to a genome draft. So you have to find the sequence, and align it with something in the genome draft. So, you might have misalignments, you might have some other things. So there are a lot of sources of noise in scRNAseq. And so it's nice to have this validation method. This is interesting. They talk about transcription rate varying across stages. So embryonic cells change markedly across stages. So when the embryo undergoes a reductive cleavage, where cell nuclear volumes decrease by about twofold each division, then we get this difference in transcription rate. So as you know, like you get cells, you have certain volume in the embryo, and as cells divide, they get smaller with respect to the embryo. So their size reduces, the daughter cell size reduces, and each round of cell division, the daughter cells tend to get smaller. So this is where, you don't need as much you don't need to have as high of a transcription rate if your daughter cells are getting smaller and smaller. When you get up to the 8-cell stage where you're founding your sublineages, you may need a lot of transcript, and the cells don't really get smaller until they get past that 8-cell stage. So once you start to get this reductive cleavage, you start to get, past the need to establish these sublineages, then the transcription rate can fall. It can lower itself. Because remember, the transcription rate requires a lot of metabolic resources. So there's something going on there too. We have to take that into account. So there are a lot of things sort of affecting transcription rate across stages. And what they did here in the paper is they found that while high transcription rates occur at the 4, 8, and 16 cell stages, the overall distribution of rates is highest at the 8-cell stage. This seems broadly true across most lineages, so this is lineage invariant, sublineage invariant, but it's just having to do with the round of cell division and the size of the cells that result. So this is really not anything that's like linked to cell or a sublineage or to what the cell type is going to be, but it's really related to the size of the cells, the daughter cells. So the median accumulation rate of medium and high rate genes is higher for cells in the 8-cell stage compared with their daughters at the 16-cell stage. Overall median rates of the 8-cell stage are about 2 to 3-fold higher compared to the 4 and 16-cell stages. So this is a nice figure that kind of shows in graphical form what we've been talking about. So again, this is the 8-cell stage here, and this is the 16-cell stage on the bottom. So the 8-cell stage is here, this third layer, and then the 16-cell stage is the fourth layer. And as you can see, you establish basic polarity up here in the second layer. You get your founder cells in this fourth layer, and then so you have your different AB cells. You have your AB sublineages. You have MS, which leads to muscle and pharynx. The E sublineage, which leads to gut. The C sublineage, which leads to muscle and epidermis. And then P3, which leads to muscle and germline. And so these are very, on the right-hand side, which is the posterior end of the embryo, you're going to get these very highly specialized sublineages. On at the anterior end of the embryo, you're going to get these very general sublineages, which produce all sorts of different types of cells. So this is, an interesting thing. You might expect to see some asymmetry here, but you really don't really see it in terms of sublineage. You see it in terms of depth of the tree. So as the daughter cells start to get smaller and smaller, that's when you see this reduction of transcription rate. And when you're establishing up to the point where you're establishing your sublineages, you have this maximal transcription rate. Okay, so we can look at this in terms of absolute change and fold change. So fold change is just in comparison with a reference where you're counting against some housekeeping gene or something that's invariant, and your absolute change is just the change of that gene in terms of the transcript count. So this is where this difference comes in for these graphs in B. You get the number of genes at log scale, so copy number decreases here with absolute change in RNA molecules. So you get a number of them that are really highly expressed with not really a long tail, but it's like you get you don't have as what they're saying here is that the very high number of copies is restricted to a subset of genes, and so that's what you might expect. That's with the absolute change measure. With the fold change measure, you have a little bit longer tail, but you still have the same pattern where you have a smaller number of genes that are really upregulated. So this is like, you imagine probably functional genes that are necessary for differentiation, for sort of establishing things in the early parts of the lineage tree and so forth. So not everything is really highly upregulated. Some things are, many things aren't. It's interesting though, as you get this change in time. So two-cell, most in the two-cell stage, which is the ABP1 stage, almost everything is low in terms of its expression. You get this medium, very small number of medium expressed genes, and this is a very small number. This is the proportion of genes, so this is like 0.25, this is about a quarter of the genes. And you can see that when you get to the 8 and 16 cell stage, at about a quarter of the genes, you start to get this medium to high regime. So this is this part of the distribution here for fold change and absolute change, this part up here. So that translates into about 25% of your genes being either medium or high, highly expressed. So, and then of course in the 8 cell, we get a little bit more in terms of very highly expressed genes than the 16 cell. And I think that decreases over time. So you get this decrease over time in the amount of upregulation. We have this other graph here, which is the number of genes, low, medium, and high. So this is breaking this out into the sublineages. So your founder cells or actually these are the A, B, E, M, S, E, M, S, C, D, and P. These are the 8 founder cells. And, this is where you're getting your the differences across the different sublineages. So you get this 2 cell here, P and AB. Then 4 cell, you start to get changes in EMS. 8 cell, you start to get changes here, and 16 cell here. And so you can see this for low, medium, and high. For high, you don't have any in the 2 cell stage. By the time you get to 16 cells, you have MS, E, C, a little bit in C, and then AB. So C and C and MS are, we go back up here, muscle pharynx and muscle epidermis. So it means that you're prepping these cells for muscle fates, but you're also prepping them for these AB fates, which include neurons and other things. So really you're, maybe some of these types of fates are where it's important to prime them with a lot of mRNAs and so forth. So this is just a heat map of the genes versus the lineage tree and showing some of these changes. So to finish up this paper, they talk about the influence of transcript decay and post-transcriptional processing on RNA output. So if we measure the RNA, we're only measuring sort of gene expression at the first stage of its transformation into something like a protein. So we get gene expression, and that tells us something about like maybe, how much transcript you need. But ultimately, we want to know what the proteins are. And so, we need to know is, first of all, the decay of transcripts over time, how much of those are going to contribute to translation. Then you also have this post-transcriptional processing, which happens, to the RNAs, so that some of them are functional, some of them are not. So it's not just enough to have a lot of mRNAs in a pool. Some of them are going to be functional or change their function for the next step, which is translation. And so, these are things we have to think about in context, and it's beyond the range of the study. But for the paper we just published on looking at gene expression proteins, it's very poorly correlated. When you look at like protein counts and you look at gene expression counts, it's very poorly correlated as to how much transcripts are contributing to proteins and their abundance. So, there are a lot of, that's because there are a lot of intermediate processes going on, and we have to be able to understand those a little bit more. And especially in something like this, to really understand their effect on, say, like the phenotype. So we used whole embryo RNA-seq data to estimate the contributions of synthesis and decay to accumulation rates, and our analysis suggests that the transcript production has a larger impact on total rates. This is consistent with studies in mammalian cell culture systems during the response to stimuli, where transcript synthesis contributes more to changing RNA levels than degradation. So this is where, you get this response to different types of things like lipopolysaccharides or hypoxia, which are stimuli that are like kind of challenging to the cell. And so this is where they have to sort of regulate their RNA levels. This is where they might overproduce certain RNAs over others to ensure that they can adapt to their conditions. And so this is where, degradation isn't the dominant thing. It's actually changing RNA levels. So different genes are being upregulated and downregulated with this stimuli. Many of the regulators that we analyze here are transiently expressed, with most transcripts disappearing within 1 or 2 cell cycles of the time of maximal expression. So this is where, we get into this issue of mRNA decay, and over time you get decay. And of course, a lot of the transcripts in this case are disappearing over just a couple of cell cycles from the time of their maximal expression. So this is an interesting thing. Thinking about like the timing of all this in the lineage tree. In accordance with this, genes with higher total accumulation rates tended to have shorter half-lives. So this is where you accumulate a lot for a gene, and they tend to have a shorter half-life. So it's kind of paradoxical. You'd think if they accumulated more genes or more transcripts, that it would decay slower. But in fact, they maybe are accumulating more transcripts because their half-life is shorter. They would run out faster if they didn't produce so much. So that kind of makes sense. If you have something that runs out very quickly, you would produce a lot of it. And if something has a longer half-life, then maybe you don't need to produce as much. However, there is a possibility for decay rates to be different during the appearance and disappearance phases. So this is something they talk about in the paper we didn't cover. Although we estimate that de novo transcription rates are modestly higher than the observed total accumulation rates when adjusting for decay, more direct measurements of half-life during the time of transcript increase versus decrease will determine more precisely how much higher and even more impressive the total accumulation rate can be. So there are a lot of things that govern transcription rate increase and decrease, and this factors into accumulation rate. So this is, something that I guess is maybe work for later or some sort of computational study or something. But in any case, they get into post-transcriptional processing as well. And of course, this includes things like RNA splicing and other types of modifications. And this is another phase of gene expression where rates may be tightly controlled. we found a number of unspliced transcripts of the MS-specific gene CEH51 outside nascent transcription sites in early muscle cells, which could mean that post-transcriptional splicing plays a role in how quickly mature transcripts of CEH51 are produced. I think this whole passage is really interesting. So the idea is that if you have a bunch of mRNAs and you splice them, when mRNAs are being spliced, they're being chopped up so that certain pieces remain functional and some pieces are taken out. And the reason you do that is because you want to create isoforms. So you have different forms of the same transcript that do sort of very specialized functional things. And these isoforms are very important in muscle, as we know from other studies. And so, think about if you are producing a lot of mRNAs and you need a lot of splice variants. So you need a lot of different types of different types of spliced mRNAs, so many different varieties of them. So you can create many different isoforms. You need to produce a lot of mRNA to do that, regardless of the decay rate, because you just need a lot of different versions of something that are going to become different specialized variants. And you need to have enough so that you can have, a pretty decent number of each. So this is interesting from the perspective of functional modifications. Direct RNA-seq is broadly revealed that many transcripts in diverse cells are not spliced co-transcriptionally, although the importance of post-transcriptional splicing and the rates of other processing steps during cell fate specification still need to be worked out. And so they also have a statement here on cell size and transcription rates. So to finish up with that, you're really dealing with cell volume and reduction of cell volume and development for individual cells. We know that transcription rates scale with cell size across organisms. The C. elegans embryo develops by reductive cleavage in a constant volume eggshell. So that means that the daughter cells get smaller and smaller, and the eggshell doesn't vary that much, so the eggshell remains constant. But as you get more and more cells, they get smaller and smaller and crowded into this eggshell, with cells reducing about twofold in size with each developmental cell division. We see the highest maximal transcript accumulation rates in the 8-cell stage, where cells are larger compared to the 16-cell stage, so suggesting that transcript rates in the C. elegans embryo may also be driven by cell size. So we have decay, we have functional requirements, and we have cell size. And so these are the three kinds of things that are controlling our mRNA production, mRNA decay, and what this mRNA balance looks like and what it does functionally. So finally, they finish up with talking about maximal achievable transcription rates. So we talked about 2 weeks ago about the speed of transcription or the speed of cell division, which is also the speed of transcription, because you need to be able to not only transcribe things, but you need to replicate DNA so that you can have 2 genomes for your 2 new cells. And so these are all interrelated. And so we have this discussion here on theoretical limits of transcription rates, talking about polymerase 2, POL2, and other transcription factor molecules, the rates of initiation and elongation, gene length, and pause sequences. So for short genes such as N3, which is around 1.3 kilobases of primary transcript, at the commonly assumed elongation rate of 1.5 kilobases per minute, the entire gene length would need to be completely occupied by POL2 complexes during maximum expression. So what they're saying is that you have these genes, you have the genome, you have these genes, they're a certain length. The transcripts are what we're really interested in, where the transcripts are sort of the things that code for the transcripts. And the polymerase 2 has to be on there, moving along, producing transcripts, mRNA out into the cytoplasm. You can't do that, it has a certain rate. So 1 polymerase 2 is a certain rate. And what they're saying is that to do this efficiently in the time required, you need multiple POL2s on the same sequence. And so what they're saying is that sometimes you get to these rates that you're predicting that require you to just have so many pull-tos that's sort of impractical for what we're dealing with. Now, we can put as many pull-tos as we want on the coding sequence. The question is that what biology is doing? Is that something that's, metabolically feasible? So that's the kind of the point that they make here. So there is a limit. There is a limit to a transcription rate, practically speaking. As some fish analysis of early zygotic genes in the Drosophila embryo has also measured high rates of transcription, potentially nearing the steric limits of pull-2, suggesting that rates approaching theoretical maximum could be a common feature of gene expression in rapidly dividing embryos. So this is where you reach this theoretical maximum related to the, how many pull-2s you can get onto a chromosome or onto a coding region of a gene, and how fast it moves, and all of that. So that's how you determine that. So this is actually maybe a common feature of rapidly dividing embryos. And so we don't know too much about the elongation rates, but we know that elongation speeds as high as 6 kilobases per minute are seen in mouse embryonic stem cells. So we know that this is possible. It's just, whether we actually can observe it. So the next thing we're going to talk about is a category called ensembles and generativity. So there have been a couple of papers that I've run across this week that have to do with the generativity of development. And then there's this paper on ensembles in development. So the first paper is this paper, The Genomic Code, and this is by Kevin Mitchell and Nick Cheney. Kevin Mitchell is a genetics and neuroscience person from Trinity College Dublin. And this is, having to do with the genome instantiating a generative model of the organism. So the abstract reads, how does the genome encode the form of the organism? And this is, of course, a question that has to do with the genotype to phenotype mapping. In other words, if we take our genotype, we produce transcripts, we produce proteins, proteins produce tissues, the tissues then form this phenotype. And so how do we encode the form of the organism? And that's, of course, something that when people started sequencing genomes, we expected to be somewhat straightforward. And of course, it has not been that. So we need to understand the nature of what they call the genomic code. And a lot of people have referred to what the genome is as a code. And of course, that's a metaphor that's fraught with a lot of problems. So they actually speak to that here, where they say common metaphors such as blueprint or program fail to capture the complex, indirect, and evolutionarily dynamic relationship between the genome and organismal form. So, this idea of a blueprint or a program, if you think about like computer programs, they're executed line by line. So you have things in there that are line by line. The code, you run through the code, you can debug the code, you can do things with rewriting the code. And we talked about this 2 weeks ago with that, some of the synthetic biology stuff we've talked about, where, people are actually doing this with bacterial cells. They're able to take a gene, they're able to rewrite the gene, they're able to knock genes out or do other things, and they can execute, different commands, basically. So you have groups that have created bacterial colonies that can produce different shapes, like, faces or patterns or whatever, or they can do things, metabolize different things on command. You can put a switch in that can be activated. So, bacterial cells, we've kind of taken the genome, but we've kind of created programs, very simple programs. The question is, does that translate to, say, animal cells? Does it translate to, say, like complex modes of development? Or, and we know we can manipulate genes, we can do CRISPR, we can do other things. But, is that sort of in spite of itself? In other words, is the complexity of those systems really not organized like a program or a blueprint? We are just able to do that because we're sticking our finger in a light socket, or is this, maybe things are programmatic? So this is an interesting question, and we don't really have the answer to that. But that's why we're talking about this, and especially this paper, because they talk a little bit more about the constructive interactive processes that produce a developmental phenotype. So such metaphors are also not readily formalized, either to treat empirical data or to simulate genomic encoding of form in silico. So this is where we want to be able to produce forms on a computer. We want to take a bunch of, say, DNA code, which could just be a series of letters or a string or a set of strings, transform that into some shape. That's what we want. We want to do that in a computer, but it's not really easy to do in a computer. So, we can do that with genetic algorithms to some extent, but it requires you to have this sort of blueprint on top of a blueprint, and it becomes pretty hard to connect to what's going on in nature. So that's an important set of points. Here we propose a new analogy inspired by recent work in machine learning and neuroscience, and that is that the genome encodes a generative model of the organism. So this is where you have the genome encoding a model of the organism that's generative. It basically is encoding all these different possibilities. And then it's, the developmental process selects from those different models or different instantiations of what's possible. In this scheme, by analogy with variational autoencoders, which is a tool, of course, in machine learning, the genome does not encode either organismal form or developmental processes directly, but comprises a compressed space of latent variables. So this is otherwise known as the latent space. And so the latent space is basically all possible combinations that you can sample from to get some sort of answer. And you sample and sample and sample, and you optimize until you get to the thing that's, most likely. So that's an interesting approach, is using this latent space to sort of get at this. These latent variables are the DNA sequences that specify the biochemical properties of encoded proteins and relative affinities between transacting regulatory factors and their target sequence elements. So this whole passage basically says that we're basically using DNA sequences in our latent space, but not every DNA sequence, just the ones that specify certain biochemical mechanisms, properties, etc. So, we're basically paring down our genome to something that's functional, or maybe, something that's relevant to a phenotype. We have all these different configurations, and then we can sample from those, and we can then select from those to get our developmental phenotypes. This is not, I don't know how to put this, but basically there's a connection here with epigenetic landscapes. So in epigenetic landscapes, we represent, all the possible forms, and then we kind of see how the developmental trajectory moves through that space. So it's almost like we're representing the genotype as this latent space, but the latent space is a landscape. And then we're moving through that landscape with a trajectory, and we're finding an endpoint where we can then say this is the outcome of development. So it's very, there's a connection there, but I don't know exactly what it is. It's just very interesting parallel there. Collectively, these comprise a connectionist network with weights that get encoded by the learning algorithm of evolution and decoded through the processes of development. So this is where we have our connectionist network, which if you're familiar with a neural network are the cells, the connections. The connections have weights. You go through different layers. Your weights are a series of connections across layers. These weights are encoded by a learning algorithm of evolution. So basically evolution tells this connectionist network, what connections are relevant, which ones are not. And then you get this decoded through the processes of development, meaning that it's then, the development is where you're getting the output and evaluating it. The latent variables collectively shape an energy landscape that constrains the self-organizing processes of development so as to reliably produce a new individual of a certain type. This is actually where they mention the Waddington's famous epigenetic landscape. So this is a direct analogy, which is this energy landscape. So this latent space shapes the energy landscape, or latent variables more specifically. So there's this sort of connection between the latent space, the energy landscape, and then this sort of selective mechanism that's, in this case, encoded in a connectionist network with weights. This is an interesting way. The way I was thinking about it more, though, is that there needs to be this feedback between selection and representation in the genome. And of course, in the last paper, we saw that we have certain genes that are upregulated due to their functional significance. And so that's something that, isn't something you can derive directly from development, just unfolding. You need to have some encoding of what these cells are going to become in order to have this upregulation of genes that are relevant for later development. So you need to have this feedback between the outcomes of development and the genes that encode development in the first place. And, I don't know if they've explicitly kind of discussed this here, but I think it's a pretty good model for getting what you want out of this. The generative model analogy accounts for the complex distributed genetic architecture of most traits and the emergent robustness and evolvability of developmental processes. And so this is where, of course, you can provide a new explanation for independent selectability of specific traits, drawing on the idea of multiplex disentangled representations observed in artificial neural systems. So finally, it offers a conception that lends itself to formalization. So we have the ability to formalize these, observations we make of development and sort of how it's generating new forms. So this is a really interesting paper. So one part of this paper, they talk about the current metaphors like program and blueprint, and those metaphors fail to provide insights on certain questions. These are the questions that people typically ask in development or that lead from those metaphors. So the first one is, how does information get encoded in the genome, and what is this information about? So what is when you just say there's information in the genome, that's not really good enough, because, what is the information? Is it just kind of like, transcribe this, don't transcribe this, stop transcribing, keep transcribing, or is there just is the entire form encoded in the genome? So this is a question that we don't really know. People throw the term code around a lot, or encode, but it doesn't mean anything unless there's actually something that's being encoded. The second one is, what is the data format of this information in the genome? So I guess what that means is like, what is the format? Is it, coordinate instructions? Is it simpler than that? Is it like migration information? Is it like, something that has to do with the collective behavior of cells? What is the data format? So this is an important point because we don't know. I don't know if it even tells you that much, because maybe it just specifies what the protein should look like. It's really not clear. How does such information get decoded through the processes of development? So even if you have information encoded in the genome, how do you decode it? How do you decode it in development so that it's useful? It's not clear. How does genotypic variation relate to phenotypic variation? So if you have genotypic variation, say if you have the splice variants we talked about earlier, or if you have a mutant form, how does that relate to phenotypic variation? Is it a direct mapping? So when you have a point mutation, do you get a specific mutation? That may hold true in some cases, but not in others. The same thing with just, your splice variants. The splice variants don't automatically map to variation functional variation. It's hard to say, if we could build a map between all the splice variants, say for muscle, and what the functions of muscle are. It's not very clear what those relationships are. So linking genotypic to phenotypic variation is hard, and I like the idea they have of using a connectionist model. I actually did something similar to that in a paper a long time ago, where we used a connectionist model to look at sort of how this variation is processed. And it's basically the idea is that you get this combinatorial mixing. So you go from different variants in the genotype, you combine different types of variation across genes, and you get these different phenotypic outcomes. But there isn't this one-to-one mapping that you can say, this genotypic variation that you see here, it relates to this phenotypic variation you see over here. I mean, it can happen, but it's very rare. And in C. elegans, you'll sometimes see, certain mutant phenotypes where a gene has a certain set of point mutations that lead to some phenotypic deficit. But that's not the norm for most of biology. You get this in specific mutants because it's very rare. That gene has a pretty large effect on the phenotype. So most genes don't have that kind of an outsized effect. How does the genetic architecture of various traits arise? So how do we even get a genetic architecture in the first place? We know there's a lot of junk in the genome. We know there's a lot of conserved elements in the genome. So a genetic architecture should be conserved, but it also should be sort of have a lot of redundancies and other types of things. So in that case, how do we get a genetic architecture that maps to specific types of phenotypic variation? And then finally, how do the underlying encodings evolve through time? So if we have an encoding that's matched to some functional aspect, if we change it, if we evolve it through time, how do we ensure that it's produces a viable phenotype? And these are all things we can't really answer with with sort of the blueprint model, nor with the code model. So the second paper here is generative models of morphogenesis and developmental biology. So this is in the seminars in cell and developmental biology. This is by Stillman and Mayor, and this is kind of following up from this last paper, talking about developmental biology and generative morphogenesis more specifically. So the abstract reads, understanding the mechanism by which cells coordinate their differentiation and migration is critical to our understanding of many fundamental processes such as wound healing, disease progression, and developmental biology, of course. Mathematical models have been an essential tool for testing and developing our understanding, such as models of cells as soft spherical particles, reaction diffusion systems that couple cell movements to environmental factors, and multi-scale multi-physics simulations that combine bottom-up rule-based models with continuum laws. So mathematical models can often be loosely related to data or have so many parameters that model behavior is weakly constrained. So we can build mathematical models that kind of describe these processes, but sometimes they're very loosely related to the real world. But sometimes we can also have so many parameters in our models because there's so many things to take into account that we can't really ground our model behavior too well. So these are all things that we have, when we use sort of precise simulations, we often need to, propose precise equations. And it's not easy to do that in developmental biology. It's easier to do that in physics. Maybe not in soft matter physics, but definitely in mechanics. But in development, doing that, it comes at a cost. Recent methods in machine learning introduce new means by which models can be derived and deployed. In this review, we discuss examples of mathematical models of aspects of developmental biology, such as cell migration, and how these models can be combined with these recent machine learning methods. So this kind of goes through some of these mathematical models and then using some of the generative techniques to sort of say, can we do things better by using these generative techniques? They're also making this analogy to development, but it's not like they're not talking about development itself being generative. They're talking about the model being generative. So in this case, they're looking at, say, mathematical modeling versus maybe something like a deep learning model where we have this generative aspect to it. So, however, as we shall describe, both modeling paradigms can be understood under the broader framework of generative modeling. And they talk about different types of generative modeling before this. Generative models seek to model the underlying stochastic procedure for generating data, opposed to discriminate models which seek to separate data into subclasses or categories. So, sometimes in equations we want to divide things up into categories or subclasses or sets or other things that are, we can use as sort of a general input. So we want to have we'll say, okay, we want these cells of this type, we want these cells of this type, we multiply them together, we integrate their trajectories, and we get this answer. With a generative model, you're not doing that. That's more of a discriminant model where you have, specific types of, you might get categorization as an outcome, you might get a predictive set of values as an outcome, those sorts of things. In a generative model, you don't get those. You're generating solutions and you're evaluating those solutions. So that's what they're getting at here. I wouldn't have really described this as just kind of a deep learning technique because, in fact, you can implement generative models with a wide range of techniques. And really, it's that difference between generating solutions and having exact solutions. That's kind of the difference here. And in fact, sometimes with differential equations, you get it's sort of generative in nature because you're generating answers. You're approximating answers and you're generating, different types of answers. So that line isn't very, it's not hard and fast. When understood as generative models, both approaches share many similarities and can, in principle, be combined. We posit that doing so would allow for interpretable models of high-dimensional data, or ultimately low-dimensional models that provide highly accurate predictions of experiment observation. So in this paper, they describe how biophysical models have been used in developmental biology, describe some of their limitations as traditional forms of generative models. We describe biophysical models of three processes fundamental to developmental biology: cell-cell interactions, collective cell migration, and physical morphogenesis. We then describe generative models that use neural network models, so-called deep generative models. We set out the main forms of deep generative models and provide recent examples of their use. As a nascent field, we are limited by the number of applications of deep generative models to developmental biology specifically. And then they talk a little bit about using traditional and deep generative methods to be combined as a form of hybrid models that both retain the explainability of biophysics and the flexibility of deep learning models. So this is where they get into the biophysics models, the interactions between cells, cell-cell interactions, collective cell migration, and morphogenesis. So here you have these different mathematical models across length scales. Cell-cell interactions are these bidirectional interactions. Collective cell migration is where you're looking at cells moving in certain directions, if they're coordinated or not. And each cell, you have to account for each cell and their trajectory and sort of a measure of, at any point in time, are they acting collectively or are they acting in different ways? There are different phases of behavior here. And then morphogenesis, where you have different components that interact, but their components vary across the model, and depending on their interactions, it can affect state changes. So they get into deep generative models here. So they talk a little bit about deep generative models. And so they talk a little about model accuracy as being evaluated by comparing model output to some specific observables or summary statistics. In this sense, our biophysical models in the last section can be understood as describing a generative process, namely a process which leads to the generation of a measurable quantity, where we measure the system or output using summary statistics. Now, these biophysical models, they distinguish this against generative models, which are distinct from discriminant models, which separate data into subsets, for example, through a classification or clustering algorithm. Instead, generative models aim to describe the entire data space and the underlying data generating process. So this is where we have, again, this latent space or this energy landscape or whatever, and then we want to take that and find solutions from that space and what the generating process is. So if we take our biophysical models and ground them in generative models, we gain insight into the main process by which a model might be developed. So they talk about implicit generative models, and they talk about this, they give two examples. One are generative adversarial networks, or GANs, and the second is denoising diffusion models. It's diffusion models and GANs, basically, that have shown extremely strong performance at generating things like text-to-image models that produce highly expressive images from text prompts. And so this is, we can map this to development of these kinds of models. So implicit models such as GANs have been used in cell biology as well for segmenting and tracking cells. And so we can do certain things like, predict 3D cell geometries from 2D images. And there's this expectation that this will grow in the future. But of course, implicit models, while, having a lot of promise, are particularly prone to limitations of interpretability, as a learned generative process is typically not straightforwardly accessible. So, just because we can sort of characterize that space and generate, outcomes, it's not easy to sort of get an idea of what the process is behind this generation, and even if we're getting the right answer. So there are a lot of problems that we might have with such a type of model in development. Then we talk about explicit generative models, and for this he uses this autoencoder and variational autoencoder example. So of course we talked about variational autoencoders in the last paper. That's what they propose for development. And so both AEs, which are autoencoders, and variational autoencoders, or VAEs, follow a bottleneck-like approach to approximating this data space. So this is where we basically take a neural network, we input data, we encode the input data into a low-dimensional space or latent space, and then decode this latent space into these different types of outputs. And so we're trying to minimize the difference between input data that is passed to the encoder and output data which decodes samples from the latent space. And so this is in variational autoencoders, we have this probability density function that we have to worry about. So we're actually building this probabilistic model that we sample from. And so this is exactly what we're talking about with the weights in the last paper. So this is really, this may allow us to build a lot of synthetic data that we might be able to exploit. But also, we may be able to build, models of biophysical properties that may not be observable. So this is another, benefit maybe to this. And then finally, we talk about these hybrid approaches. So to develop models directly from data, explicit generative models can be combined with methods for inferring symbolic equations. And so this is where we have this ability to synthesize symbolic equations or, build symbolic equations for systems, but also have this generative model where we're producing different possibilities. So there's a whole literature on sort of producing scientific laws and people applying machine learning techniques to scientific data sets to find candidate laws. And those laws are described as equations. So this is an area that's, people have been revisiting this for decades and not making some progress, but it's been kind of rooted in physics mainly. So, having a role, for this in developmental biology is really exciting. So a very specific example here are reaction diffusion systems, where we have only partial model equations, and we can use artificial neural networks to augment model equations where only partial dynamics are known, but a full model output is available. So this is something that's exciting in terms of like getting, connecting the equations with these outputs. Overall, hybrid learning resolves the tension between modeling paradigms by restricting the application of neural networks to a subset of the broader biology under investigation. Finally, we have this paper from Stuart Kaufman from Physica A. And the title of this paper is The Ensemble Approach to Understand Genetic Regulatory Networks. So they don't get into regulatory networks too much in the last two papers, although the first paper, I think that's the direction they're going. This talks about understanding genetic regulatory networks, which are often kind of opaque in terms of how they map to actual biological systems. But what Stuart's going to do here is give us an ensemble approach to this. So the abstract is very short, and it reads, understanding the genetic regulatory network comprising genes, RNA, proteins, and the network connections and dynamical control rules among them is a major task of contemporary systems biology. I focus here on the use of ensemble approach to find one or more well-defined ensembles of model networks whose statistical features match those of real cells and organisms. Such ensembles should help to explain and predict features of real cells and organisms. A couple things to say about this paper. First of all, he's made attempts in the past to use random Boolean networks, where a gene is modeled as if it were an on-off device. So your network has n genes with k inputs. So you have a certain number of genes. Each of those genes have a certain number of inputs. And the time is updated synchronously by a central clock. So basically, this is not something that you see in cells. This is just kind of an abstraction where you have a certain number of genes, a certain number of inputs, time component, and then this switch that goes on or off. So this, may be useful in terms of understanding very simple networks where, you're turning on genes or turning off genes. Like in the first paper where we talked about, building an mRNA pool. You might be able to do something like that. But beyond that, maybe it's not so good, for example, to understand fine-tuned regulation of specific traits or something like that. And then at least 3 initial ensembles of Boolean networks distinguished by their wiring diagrams are now available. So we have these classic random Boolean nets, where each of the n genes receives the same number, k inputs, randomly chosen among the n's. So you have these random nets where the inputs vary across genes. You have these scale-free networks in which there are power law distributions of inputs and outputs, or both from the genes. So you just have a distribution of inputs and outputs that follow a power law, where you have some genes with many inputs, some genes and outputs, many genes with many fewer inputs and outputs. And this is sort of this, there's no specific, scale to it. It's just kind of in that, according to the tendency. And again, that's useful, again, for some of the things we saw in the first paper, where we saw kind of what looked like they could be power law distributions of the amount of transcript produced by a certain number of genes. Then we have these Medusa networks, in which a small regulatory head, or like maybe 1 or 2 units that are regulatory, contain a network among transcription factors that regulate one another, and an acyclic directed graph hanging off that head, which contains genes that are regulated but not regulating. So you have like genes that are regulating each other in these small modules, you have a head or a couple of heads that do the regulating of these modules, and that's how, it's kind of like a very highly modular network. The sparse data available tend to support either a scale-free network, and this is true in E. coli, or a Medusa network diagram, which is true in yeast. So these are citations 11 and 13. So basically, you have examples from E. coli and yeast, which tend to suggest that it's either scale-free or Medusa. In addition to these structural classes, different biases in the classes of Boolean functions can be introduced, particularly including canalizing and high P functions in certain post-classes. Beyond Boolean nets, one can consider discrete S-state networks that remain synchronous, Boolean or S-state networks that are randomly asynchronous, probabilistic Boolean nets, Boolean nets in which genes have a distribution of timescales, networks of piecewise linear equations, networks with continuous cell functions. So these are equation-based networks or governed by equations. Networks with more detailed and realistic equations representing both RNA and protein synthesis. And then finally, networks governed by stochastic equations of motion. So the ensemble approach can include all these different types of genetic networks. The Medusa networks are of particular interest in that evolution can add new genes to the acyclic graph of genes that are regulated by the head. So you have this, basically this sort of, ordered set of connections that get regulated by a head. And so the head actually serves to regulate the entire set of genes that are connected in a certain direction. So we have this evolution can add new genes but plays no regulatory role without altering the attractors of the regulatory head. So this is where evolution is acting on the network merely in terms of selecting the components, not the regulation itself. The regulation can emerge from the interactions. Okay, so I think that was a fun tour of those papers. Hope you learned something.